Thank you all for being here. Thank you, Sean, for the invitation. It's an amazing turnout. It's really fantastic to see this. In 2008, when I started studying the ketogenic diet, it was primarily for pediatric epilepsy. So when you put it in Google, the Charlie Foundation came up, which was Charlie Foundation for Pediatric Epilepsy, and now it's the Charlie Foundation for Pediatric Epilepsy and other things. So it's fantastic. I always thought the low-hanging fruit really was obesity and type 2 diabetes, and it's been amazing. It's been incredible to see the scientific research and the interest in that grow over time. So I'm really excited today to share with you really a story that goes back maybe about 20 years ago. As a neuroscientist and doing my PhD at Rutgers, I was primarily focused on patch clamp electrophysiology. I was focusing on brain energy metabolism and the basic science that really was the foundation of what I was doing, the neural control of autonomic regulation, and as it pertains to extreme environments, low oxygen and high oxygen, and different changes in pressure. That research ultimately led me to ketogenic nutrition as a countermeasure, as a metabolic sort of therapy and strategy to enhance performance and performance resilience in extreme environments. And now we study many different things, many different emerging applications came out of that original research. See if I can get this to work. Okay. Before I begin, I really want to thank our sponsors. And the sponsors for our lab really make it possible for me to do what I do. And we have sponsorship from federal organizations like the Office of Navy Research, the Department of Defense. We partner with NASA on different projects. NAVSEA is sponsoring a project right now at Duke University where we're using the ketogenic diet in humans and pushing them to the edge of seizure. And we want to look at various physiological biomarkers that will predict an impending seizure and do that with and without the ketogenic diet. So we also have industry partners. I have internal support through my university, through our patents and licensing, and also the foundation. And we work with a number of small foundations, 501c3 foundations, for a number of different rare disorders and also cancer research. So the information that I'm presenting to you today is not to be taken as medical or nutrition advice. So I'm a PhD scientist, a basic scientist. We do work with medical doctors and nutritionists. We're working, today my lab is working with the Moffitt Cancer Center on helping to develop a therapeutic protocol that could be used for cancer. But my advice today will not be sort of a prescriptive advice. And so my university also wants me to kind of say that I'm the inventor on a number of patents that have been licensed to various licensing partners, and the royalties from those patents, from the products that have been commercialized, actually go back to support our lab. And some of the royalties that we've gotten from various products that are out there has actually helped fund various undergrad students that are now in medical school, and some of them are moving on, and they are really carrying the message forward. So that's been inspirational to see that. And I have a number of conflicts of interest related to being an inventor on various patents, and various use patents and composition of matter patents that are out there. I'm also a co-owner for Ketone Technologies, and Ketone Technologies partners with government institutions, in particular NASA, to help develop sort of resilience strategies and looking at cardiometabolic measures and looking at things like sleep in extreme environments. So we help to fund some of that research. And today, my talk is really going to be on kind of how I got started in studying all this. And that was, like I had mentioned, environmental stress. And that's looking at the effects from the level of the cell to the tissue level, to the whole animal level, and now we do human studies on manipulating oxygen and pressure from low oxygen to, that would correlate to altitude or the space environment, or high levels of oxygen, which would correlate to the hyperbaric environment that the Navy SEAL warfighter would be exposed to. And I'm going to transition into talking about how ketosis could be used as a countermeasure to enhance resilience, performance resilience in these extreme environments, and just talk about the different methods of ketosis. So there's fasting-induced ketosis, there's dietary ketosis, and a big part of what I'm going to talk about today is actually supplemental ketosis. So using ketones as an energetic source. I think of them as food, as the fourth macronutrient. They're calorie-containing substances that can be integrated into the food or given as a bolus to acutely produce a state of ketosis that would mimic a fasting condition that would take you 7 to 10 days to achieve without eating anything. So there are practical benefits to using exogenous ketones, and I'm going to cover a little bit about that, and also some therapeutic applications. And the applications are really growing tremendously. As I mentioned, pediatric epilepsy was really the primary application for the ketogenic diet, and that goes back about 10 to 15 years ago when I got interested in this topic. And I'm going to cover the proven applications of the ketogenic diet, and that's primarily human studies, and also talk about what I'm kind of really excited about is the emerging applications of the ketogenic diet. And that would be a number of studies that we've done in animal models and also some small human studies, pilot studies. So there are a number of different ways that you can monitor your level of ketosis. So that would be urine ketones, breath ketones, and blood ketones. And also we use something called the glucose ketone index as a single measure biomarker, as a therapeutic sort of biomarker. And I'll talk a little bit about that, and also just finish up with a summary of some of the ongoing and future studies. So it goes about 20 years ago, my primary focus was neuroscience and doing brain recordings using something called patch clamp electrophysiology. And we would record directly from neurons in the brain, or in primary cultures, or in brain slices. And we would look at the effects of low oxygen on these cells. And I was studying an area of the brain called the rostral ventral lateral medulla, and it controls our physiology. And so the physiological responses to hypoxia varies, and the brain, various brainstem mechanisms control that. So I looked at the oxygen sensing neurons in the brain. And as I moved on with my research, we moved patch clamp electrophysiology and also atomic force microscopy and confocal microscopy. And we adapted these forms, these technologies into environmental chambers. And these environmental chambers allowed us to look at reactive oxygen species and also the membrane potential of the cell. So we saw that under different conditions, the relative level of oxygen here, as the relative level of oxygen increases, that increases reactive oxygen species. And the reactive oxygen species are coming from the mitochondria. And with the various technologies like atomic force microscopy, we could determine that the membrane surface was being altered. There was membrane lipid peroxidation and also protein aggregates that were forming in response to high levels of oxygen. And we kind of predicted, or my theory was that this was under metabolic control, that the resilience of these cells under extreme conditions were really a consequence of maintaining the ATP levels in the cell. So I was manipulating glucose, and I saw that when I elevated glucose levels, I saw a lot more reactive oxygen species and a lot more cellular damage. And then when I decreased glucose or gave a glycolytic inhibiting drug called 2-deoxyglucose, I could reduce the oxygen-induced increase in superoxide anion. So this is a brain slice preparation, and this is the hippocampal CA1 region of the brain. And we would put these brain slices into an in vitro sort of bath solution, and we would manipulate the environment to stimulate a Navy SEAL dive, for example. And under high levels of oxygen, we would see these cells here, and there's hundreds, if not thousands of cells here, would dramatically increase in the level of reactive oxygen species. And if I decrease the amount of glucose, that would be significantly lower here. So using the same hippocampal brain slice preparation, we would record the brain slice itself has an intact cytoarchitecture. So you can stimulate in one region and record from another region. And under conditions of hyperbaric oxygen, like high levels of oxygen, that would simulate about a 120-foot dive underwater, breathing pure oxygen. If we stimulate on one side of the slice and record from another, we would see an increase in the amplitude of the orthodromic population spike. So all the neurons firing together. So the neurons were really hyperexcitable when experiencing this high level of oxygen. And also we saw, with a single stimulation, we saw the occurrence of these secondary spikes. And the secondary spikes are actually analogous to a seizure in a slice. So you stimulate one time, and you see a large amplitude burst in the neurons firing in synchrony, and then you see multiple spikes that's analogous to a seizure. So this preparation actually allows us to study seizures in a slice. So we gained a lot of insight into manipulating oxygen and also manipulating the metabolic substrates. So at the time, I was personally interested in something called alpha-L-polylactate, and it was an ingredient in a supplement that I was using. I was mountain biking like 20 years ago, and alpha-L-polylactate was used in a product, I think Cytomax, and I was looking at lactate, I was looking at various amino acids and manipulating glucose and looking at the resilience in this preparation. So ultimately, we moved to rat studies where we have a system where we can monitor EEG, diaphragmatic EMG, and also EKG. So we get heart rate activity, we get respiration, and we get various EEG leads. And doing these studies, we dive the rats to 5 atmospheres of oxygen, and within 5 to 10 minutes, they typically have what's called a tonic-clonic seizure. And the seizure quickly stops when we flush out the oxygen and put in air, even hyperbaric air, and then we depressurize. And that stops these tonic-clonic seizures. There are various things that we don't know how to predict or prevent these seizures. And these seizures are a limitation for Navy SEALs that use a closed circuit rebreather. And these seizures are also a limitation of hyperbaric oxygen therapy. So if you, for example, get carbon monoxide poisoning or have an ischemic wound or decompression sickness from recreational scuba diving, you get put inside a hyperbaric chamber, and those protocols are dictated by preventing oxygen toxicity seizures. You can only go so high. So what we have shown in our rat model is that there are early predictors in respiratory control and even heart rate variability. So you can imagine wearing some kind of technology that actually tells you you're about to have a seizure. And we think that's the case with epilepsy too, or certain forms of epilepsy, the various respiratory rhythm generation and heart rate variability changes just prior to a seizure. So more recently in the last couple years at Duke University, and this is a project funded by NAVSEA, we actually instrument humans with recording device, EEG devices, where Richard Moon's lab, environmental neuroscience lab, or environmental physiology lab, takes these subjects and pushes them to the edge of having a seizure. And they do it on a standard diet, and they do it with subjects that are in a ketogenic diet. And this study is ongoing, and it's blinded, and we don't know all the results yet, but I think it's going to be some interesting findings. And in addition to these human studies, we partner with NASA as part of a Mars analog. So NASA has 14 different space analogs, and one of the analog is NASA Extreme Environment Mission Operations. And NEMO 22 was a mission that was in 2017, and I was a crew member, and it's the only sort of analog that uses astronauts. So it was really amazing to work with these astronauts. And in the mission, we do probably 15 to 20 different objectives. And one of the objectives is actually looking at cognitive function, looking at reaction time, looking at decision making. And they use various devices, including an iPad that you can use underwater. But also, this iPad has software called Joggle, and Joggle is used on the space station. And it really gives insight into how you are functioning cognitively, reaction time, decision making in these extreme environments. So we're collecting data on multiple missions. And then on a standard diet, my standard diet was a ketogenic diet. So I was on the ketogenic diet, but in future studies, as we collect more data, we're going to have the aquanauts. If you're underwater for more than 24 hours, you become an aquanaut. And many astronauts, about 60 or 70 astronauts are aquanauts. So they do a lot of their training underwater in the neutral buoyancy lab at NASA, but also in missions like this. So we can collect data from crew members in and out of ketosis. And we're looking at things like the gut microbiome. We're looking at sleep. We're looking at cognitive function, as I mentioned, body composition alterations, and metabolic, cardiometabolic parameters. So C-reactive protein, we're looking at triglycerides, cholesterol, everything you can think of. So we're collecting a lot of data, and because the sample size is small, there's only 6 crew members. There's 4 astronauts typically and 2 habitat techs. As we move along, I think it'll be exciting to see how nutrition is really impacting our resilience in these environments. And I have some interesting data on myself that we're working on putting this into a publication. But as it pertains to CNS oxygen toxicity, there are really just a few countermeasures that the Department of Defense or the Office of Navy Research has. And the main countermeasure is just to stick to the dive tables, or your dive watch will tell you how long you could be in that environment at a certain partial pressure of oxygen. But it's not always feasible for a Navy SEAL to limit their exposure to the undersea habitat, especially if you're being shot at. So you don't want to surface, so it's not always feasible to surface under certain conditions. So that's always and your individual susceptibility to oxygen toxicity varies tremendously. Even in the same person could experience a seizure one day that's only a few minutes relative to a couple hours. So it really depends on your physiological state and the state of your brain. So that varies. And the dive tables are very conservative and will limit that, but not always. So another big project that the Office of Navy Research had was looking at antioxidants. I demonstrated or showed you some data that there's an oxygen-induced increase in reactive oxygen species, in particular superoxide anion, which is the precursor to other free radicals like hydroxyl radical. So I spent a lot of time and effort, and there have been many people funded looking at antioxidants, and antioxidants tend to work really well in vitro preparations, but they don't work well in vivo models, animal models, and humans. So that's kind of been put on the back burner, and as of now, the drugs by vigabatrin, or I like to call it vigabatrin because it reminds me of GABA. So it's actually a GABA. It's a substance that actually increases the activity of GABA in the brain to very high levels. The problem with this anti-epileptic drug is that it works to a certain degree. You get about a 200% delay in the latency to seizure, but anti-epileptic drugs cause side effects like lethargy. This particular drug actually affects the eye, and it can decrease your field of view and even make you blind. So this is really the go-to drug that they have right now is vigabatrin. It's typically sort of something that would be used in a disabled sub scenario. So if a submarine is disabled on the bottom of the ocean floor, you need to denitrogenate very quickly by breathing oxygen, and astronauts actually denitrogenate before they do an EVA. So you need to denitrogenate as fast as possible with oxygen, and you could potentially have a pretty severe seizure before doing that. So the drug that would be used right now is vigabatrin, but there is really compelling data that showing that fasting ketosis can actually delay the latency to seizure. And this was work done by the Israelis a long time ago. Actually, the Germans and the Austrians did an incredible amount of environmental neuroscience and environmental physiology. And the Israelis also have contributed tremendously to this. So it kind of got past my radar, but in 2007, 10 years after it was published, I saw this study that was replicated in another lab that fasting an animal, a rat in this case, 24 or 36 hours, extended the latency to seizure by 250%. So that was about as good as any anti-epileptic drug, even at high doses. So I became really increasingly fascinated as to how changing brain energy metabolism with fasting could delay seizures. And I've shown this slide so many times, and sometimes I always want to take it out, but I can't take it out because it's so good. It's an amazing study. So it's the point that I want to make is that, and the point that was very clear in studying fasting and anti-seizure effects, is that shifting your metabolic physiology fundamentally changes your brain energy metabolism. And it also changes the neuropharmacology of your brain. So neurotransmitter systems change. Like so many different things I'll talk about later change from changing your metabolic physiology. So that really dictates what your brain is being fed, right? So in a fasted state, I mean, it was assumed prior to 1967 that 100% of brain energy metabolism came from glucose exclusively. There was some studies done by George Cahill and the postdoc at the time, Oliver Owen, and I was fortunate enough to actually connect with and talk with Dr. Cahill before he passed away in 2012, and he had some remarkable stories to tell. And one was this, study that was published showing that they fasted subjects for 40 days. So no calories, they gave them electrolytes and water, but no calories essentially for 40 days. And in that state, after about day 10, the level of ketones actually exceed the level of glucose in the blood. And at about this point, about 2/3 of brain energy metabolism is coming from ketone bodies relative to glucose. So there are very powerful homeostatic mechanisms that maintain our blood glucose levels. So that's why glucose essentially does not go to zero. He didn't measure insulin here, but insulin goes down quite a bit. Other people have measured insulin in a fasted state. But note that the ketone body, beta-hydroxybutyrate, is elevated above the level of glucose, and ketones essentially freely pass the blood-brain barrier to provide energy for the brain. So through the monocarboxylic acid transporter. And so there doesn't need to be any really keto adaptation. So you could give ketones, and the ketones will readily cross the blood-brain barrier and get into the brain. We do think that with keto adaptations, there's an upregulation of the transporters and various ketolytic enzymes that allow the neurons to use the ketones for fuel. So there's definitely keto adaptation takes place physiologically and also in the brain too. But an important point is that your brains can always use ketones for fuel. In a remarkable extension of this study, when these patients were fasted, when they were experiencing this starvation state, they were injected with insulin. 3 of them were injected with 20 IUs of insulin, which actually brought the glucose down to about 1 millimolar, 1 to 2 millimolar, around this range, which is universally fatal if you don't have your ketones elevated. So a very dangerous hypoglycemia. I teach it to medical students, at about, if you go below 2 millimolar, you start to enter a coma and death can come shortly after that. That's called hypoglycemic shock or insulin shock. So these patients, they didn't have brain damage. They were actually asymptomatic for hypoglycemia when they were given 20 IUs of insulin. So it was a dramatic demonstration that the brain is incredibly metabolically flexible and can adapt from using glucose to using ketones for a fuel source. So your brain does not use fatty acids for fuel. They're abundantly available, but they don't cross the blood-brain barrier. So your brain converts these fatty acids to ketone bodies, beta-hydroxybutyrate and acetoacetate. So these ketone bodies really are energy metabolites, and they're also signaling molecules. So more recently, and we'll hear more about it during the course of this conference, is that ketones have remarkable signaling effects in many tissues, including the brain. And our lab is kind of brain-centric, so we have a lot of tissue that we're looking at now, and we're looking at various enzyme systems and transporters, and the brain changes remarkably in response to being exposed to ketones, whether that be a fasting or the ketogenic diet or supplemental ketones. Being in a state of ketosis is really clinically happening, if you will, when your blood ketone levels are above 0.5 millimolar. And that typically happens if you stopped eating today. After about 24 hours of not eating, you start entering a state of ketosis, typically fasting ketosis. But it's kind of a difficult state to achieve and even more difficult to maintain. And I'll talk about that. Urine ketones typically have to get above 15 milligrams per deciliter. Nutritional ketosis, I will define as a strategy that elevates blood ketones. And sometimes I lump in various ketogenic agents like medium-chain triglycerides. There are various ketogenic fats that when incorporated into your diet can put you into a state of ketosis. When I speak to at scientific conferences or even the medical students, they always bring up diabetic ketoacidosis or ketoacidosis in general. So this is a physiological state where your ketone levels are very high. It's typically a state mostly associated with insulin insufficiency associated with type 1 diabetes, where you have high glucose and also high ketones. And that creates a very dangerous acidotic condition. And if it's not corrected with insulin, you will succumb to a coma and death. So generally speaking, normal physiology does not really promote going into a state of ketoacidosis. Under certain conditions, it can. There's like alcoholic ketoacidosis. If you drink too much alcohol, it impairs hepatic gluconeogenesis, and then you could have runaway ketogenesis and alcoholic ketoacidosis. So there's certain situations that we teach in med school, but generally speaking, you do not go into ketoacidosis if you're not type 1 diabetic. It happens sometimes in kids, but it's pretty rare. So keto adaptation is a time-dependent physiological shift towards using fat and ketones for fuel. And I want to stress that it is really time-dependent because we're seeing now that there are remarkable adaptations over time. Your body can acutely use ketones as fuel, but ketones have so many different signaling properties, even changing various gene expression profiles that can alter tissues in your body, skeletal muscle, your liver, and also your brain. So keep that in mind that keto adaptation is a process. And exogenous ketones are really what I'll be focusing on in the next couple slides, and these can be produced synthetically or naturally and derived from a number of different substances. They can rapidly induce ketosis and sustain ketosis if you just give a ketone salt, you can induce it, but you're not going to sustain it. If you take a ketone salt preparation and mix it with long-chain fats or medium-chain fatty acids, you prolong that pharmacokinetic profile so you can get your ketones elevated for several hours. And with ketone esters, if they're administered, you can get your ketones elevated all day with 1 or 2 doses. So this is what we call therapeutic ketosis, and it has legitimate applications I'll talk about. So we know that ketosis can be induced with fasting, with following a macronutrient profile that's part of a ketogenic diet. The classical ketogenic diet was like 90% fat. The modified ketogenic diet was advanced, or the modified Atkins diet was advanced by Eric Kossoff and John Freeman at Johns Hopkins, and that's sort of the diet that I personally follow, and it's also used for adult epilepsy. So following these strategies essentially depletes liver glycogen and suppresses the hormone insulin. It's really important to acknowledge that suppressing that hormone insulin drives hepatic ketogenesis in the liver, the production of these ketone bodies, beta-hydroxybutyrate, at the bottom, acetoacetate, and spontaneous decarboxylation, which occurs naturally with acetoacetate, produces acetone. And you blow off acetone in your breath, and that's the fruity smell that you might smell sometimes in people who are in a state of ketosis, especially high ketosis. So ketones are alternative energy substrates that are used by the brain, the heart, and the skeletal muscle. The skin also uses ketones. We know we're doing wound healing studies showing an enhancement of blood flow to the skin, an enhancement of cellular repair in the skin. So you can largely circumvent the dietary restriction typically associated with getting into ketosis with the use of various ketogenic fats like medium chain triglycerides and also exogenous ketones. And these include ketone electrolyte preparations combining a ketone body, beta-hydroxybutyrate, in some cases acetoacetate, with a monovalent or divalent cation. So sodium, potassium, calcium, and magnesium can ionically bond with ketones, and these can be formulated and ingested, and you liberate the ketones and the minerals in your body. They need to be formulated in such a way that you don't have a mineral overload. So it can't all be potassium, and it can't, if the sodium's too high, you can get sodium overload. So these need to be formulated in very strategic ways, and we do a lot of this formulation in the lab. So ketones are also signaling molecules. So my colleague, Dr. Jung Roh, really got me into this in 2007 or 8. He turned me on to using therapeutic ketosis for CNS oxygen toxicity. I had sent him an email, and he said, let's talk. And he felt that inducing ketosis could ameliorate CNS oxygen toxicity. So he was sort of the first person I connected with, and then I connected with NIH and Oxford and a number of people who were developing various exogenous ketones. And the state of ketosis, that could be fasting, the ketogenic diet, and now we know supplemental ketosis, literally fundamentally changes the neuropharmacology of the brain. And I don't have time to go through all this, but some of the things that we are focusing on are reduced oxidative stress when ketones are elevated. So they have a remarkable effect. I showed you the CA1 hippocampal region, and we also do primary dissociated neurons. If they're exposed to just 2 to 5 millimolar of ketones, that dramatically decreases oxidative stress and the production of superoxide. A reduction in glycolysis. So there's a drug called 2-deoxyglucose, and some investigators are using this in NIH-funded work and Veterans Affairs funded work as the ketogenic diet in a pill. And it does not elevate ketones. It actually turns off glycolysis or turns down glycolysis. So we're using this drug in our cancer studies. So simply reducing glycolysis has anti-seizure effects. Exogenous ketones, when given acutely, dramatically lowers the level of glucose, and I'll show that in another slide. So it does reduce glycolysis, and by reducing the hormone insulin, insulin drives glycolysis. So the ketogenic diet, by suppressing the hormone insulin, stimulates ketone production, but that suppression of insulin also reduces glycolysis, so that has anti-seizure effects. The GABA to glutamate ratio changes considerably. Glutamic acid decarboxylase, the GAD 65 and 67, are dramatically upregulated with the ketogenic diet, and they're also upregulated with exogenous ketones. So it helps convert a hyperexcitatory neurotransmitter, glutamate, to GABA, which is more of a brain stabilizing neurotransmitter. So we're doing a lot of work now looking at dopaminergic systems, serotonergic systems, GABAergic systems, and GABA really comes out of our data. Every study that we do, GABA is higher. So adenosine is also elevated. We don't really know why. The cells tend to pump out a lot of ATP, and this ATP breaks down to adenosine. And adenosine has remarkable effects. It's neuroprotective. It also has a remarkable vasodilation effect. So brain blood flow increases about 30 to 40% when you acutely give ketones. And we think that vasodilation could be associated with adenosine A1 receptor signaling. So we're studying that right now. Acetone goes up. So acetone, we think of nail polish remover, but acetone actually has remarkable functions in the body subnarcotic levels of acetone. High levels can be very destructive, obviously, but acetone actually opens potassium channels. If you open a potassium channel in the brain, you hyperpolarize the resting membrane potential, and that decreases action potential frequency and calms the brain down. So we're looking at a big focus of what I'm doing is looking at acetone as actually like a neurotransmitter. There are various ketone receptors, G-protein coupled receptor 109A, GPR43, GPR41, I believe. There's an acetoacetate receptor. So there's these receptors now, and when they're, the ligand is ketone body, and the ketone body, when it interacts with this receptor, has remarkable effects, hormone-like effects, neurotransmitter-like effects, at reducing inflammation, for example. So we also know that ketones have histone deacetylase inhibitor activity, and a big focus of what we're looking at now is actually the anti-inflammatory effects of ketones. And through the NLRP3 inflammasome, our collaborator, Deep Dixit at Yale, I constructed the diet for him, and he ran it in a mouse model and showed that beta-hydroxybutyrate reduces the activation of NLRP3 inflammasome, which is kind of like a hub that, when activated, stimulates many inflammatory processes in the body. And we're focusing sort of on that pathway too. But seizures can be activated by neuroinflammation, and seizures beget seizures. And if you have a seizure, even independent of the etiology, it can trigger neuroinflammation. So we're doing various studies now where we're given LPS, and LPS stimulates seizures in a variety of models. So we're inducing a state of ketosis and suppressing that inflammation. And we have some nice data coming out showing that you can suppress, by virtue of suppressing inflammation, you can suppress seizures. So we think some of the effects, some of the anti-seizure effects of the ketogenic diet are linked to the anti-inflammatory effects. So this is a new direction our lab is going in right now, and it's a pretty active area of research. So when it comes to applications, as I mentioned, when I got into this, it was pediatric epilepsy. So I really felt the low-hanging fruit was weight loss and also weight maintenance. It's relatively easy to lose weight, but keeping that weight off, I think, is where the ketogenic diet really shines, and or just simple carbohydrate restriction. There's human data on type 2 diabetes now, and we study a variety of sort of rare metabolic diseases like inborn errors of metabolism, and that includes glycogen synthase disorder type 2, or Pompe syndrome, multiple acyl-CoA deficiency syndrome, glucose transporter type 1 deficiency. There's many different diseases where really the standard of care is nutritional ketosis. PDH deficiency, for example, like the standard of care really is nutritional ketosis. So ketones, supplemental ketones, can really be a magic bullet for some of these rare diseases, and that's kind of why I'm focusing. Emerging evidence, and this would be animal model data, and also some human pilot studies. Probably have to update my slide, because I think even type 1 diabetes is becoming sort of a disease that could be managed with a carbohydrate-restricted diet. My student, Andrew Kutnick, is type 1 diabetic, and he gave a TEDx talk talking about managing type 1 diabetes with a low-carb diet. And I think within 5 years, there's a number of different things here. Some you've heard of, some maybe you haven't. When it comes to cancer, cancer is kind of like a hot topic. I think within 5 years, you'll see cancer moving over to an accepted therapeutic application. And when I started this, I think there was 2 clinical trials with the ketogenic diet in cancer, and now there's 31. I just checked a few days ago. There's 31 registered clinical trials on clinicaltrials.gov using nutritional ketosis, the ketogenic diet. In most cases, about 2/3, it's as an adjuvant to the standard of care, and that's what we're kind of focusing on too with the Moffitt Cancer Center. So the neurological applications are also expanding dramatically. I don't have time to go through all of them, but we're looking at things like Kabuki syndrome. With Kabuki syndrome, the ketones are actually functioning as an epigenetic therapy. So our collaborator at Johns Hopkins demonstrated that the ketone body beta-hydroxybutyrate influences DNA methylation, and it silences these symptoms in a mouse model of Kabuki syndrome. So we have a big project now that we're studying. We have the Kabuki mice that we're breeding up, and we're giving them ketones, ketogenic diet, various ketone supplementation protocols in addition to that. So I never thought that we would be studying ketones as an epigenetic therapy. So that's a pretty active area of our research now. And Angelman syndrome, that's another. I had a PhD student that studied ketone esters in an Angelman syndrome mouse model, and it had remarkable effects, better than any drug that has ever been tested that I know of for Angelman syndrome. So now that mouse model research has now led to a clinical trial on clinicaltrials.gov, and you could see ketone supplementation is being used for Angelman syndrome, and they're recruiting patients for that. So I'd like to talk about all these, but I don't have time. But anesthesia resistance is like a new application that we saw. Just by watching the mice and putting them under anesthesia when they're in ketosis, it took like 50% longer to 30% longer. So we were like, we got to study this. So we recently published using isoflurane, but we think it also holds true with carbon dioxide narcosis, with carbon monoxide, and also nitrogen narcosis. So we're breathing 80% nitrogen now, but if you're at 10 atmospheres of pressure, the nitrogen that you're breathing becomes narcotic. It's called narcosis, right? Nitrogen narcosis. So we think ketones really help with that. So that has various obvious operational implications there. And then, of course, CNS oxygen toxicity, which is what I study. In 2000, well, we started this, in 2009, I think, but in 2013, we published the first study, to my knowledge, that simply giving ketones to animals that were eating a high carbohydrate standard diet, we would acutely put them into a state of ketosis. In 15 minutes, it would look like the blood profile would look like you fasted for a week. So we used a very powerful ketone ester for this. How we did it, we have rats, and we would gavage them, intragastric gavage them, water or a ketone supplement. And we would collect data under air conditions and also 100% oxygen, but not at pressure. So normobaric, 100% oxygen. And we would look at diaphragmatic EMG, heart rate variability, and also EEG. So typically that would all stay normal. If they were gavage water, what we saw, within 5 to 10 minutes, they would have a tonic-clonic seizure. Typically, always. We've never found a rat that didn't have a seizure. So you can't avert oxygen toxicity. When we gave them a ketone supplement in the form of 1,3-butanediol acetoacetate diester, which elevates ketone bodies, beta-hydroxybutyrate and acetoacetate into starvation level ketosis. So the subjects that were in the Cahill study that fasted for 40 days, that level of ketosis in 30 minutes. And we would put the animal in the chamber and give them 5 atmospheres of oxygen, and they would have resilience up to 60 minutes. So if this animal here was gavage water, this is at 60 minutes. Within 30 minutes, that animal would have been dead. So this is double that. This is 60 minutes, and we've shown in every single animal that we dove, we had never seen that with drugs or antioxidants, that it delayed the latency to seizure. So we published this, and then after that, there's been 10 different animal model studies giving ketones, independent of diet, just giving ketones. And it was given intrathecally, IV, and various preparations. We view ketones as a 4th macronutrient. So they are calorie-containing energetic substances that can be incorporated into food. And when you eat that food, it could be a high carbohydrate diet. It could put you into a state of therapeutic ketosis. So and we also do intragastric gavage, which is acutely inducing ketosis. So the last 4 studies here, so this is the hyperbaric oxygen study on top, and we have colleagues, on the bottom is the Angelman syndrome study, where we incorporated 10% of the food by weight was a ketone ester. And that increased, everything from long-term potentiation in the brain to behavioral modifications, elevated plasmase, there was less anxiety and less seizure activity, and increased motor function activity too. So there's very active research going on right now on the seizure front. I do a lot of that. I think what may be most relevant to this group here is probably the most remarkable effect that we've seen with exogenous ketones is that when you induce ketosis, it has a dramatic glucose lowering effect. So we do lots of research with metformin, the drug metformin. Even when we use very high doses of metformin, we get nowhere near the glucose lowering effect that we see with exogenous ketones. And that includes things like 1,3-butanediol, ketone salts, ketone esters, even medium chain triglycerides. If you elevate ketones with MCT in a big bolus, you can lower glucose levels. So the study on the upper left-hand corner was actually taken from a study in International Journal of Cancer where we integrated 1,3-butanediol and ketone ester into the food, 10% by weight, and we fasted them overnight. Actually, it was just 8 hours, and then we exposed them to the food. And as they ate the food over the course of 12 hours, you could see that the blood glucose goes down very remarkably. So it's decreasing hepatic gluconeogenesis. We feel that that's happening. Perhaps, I know Dr. Richard Beach thinks it's an enhancement of insulin sensitivity. I don't know if it's that, but we are studying this right now, and what we've observed, we have some data in humans too, and the bottom shows ingestion of ketone ester, 30 grams, and you can see the acute lowering of blood glucose, about 20%. We published some of the data in rats, and we use a variety of different ketogenic agents. And one thing that always kind of stands out is that there's this correlation that if you raise ketones, you get a corresponding decrease in blood glucose. So they're inversely sort of proportional. So we have a lot of work, a lot of additional studies that we'll be publishing pretty soon on that. So my wife's a behavioral neuroscientist, and she observed that when we were gavaging the rats prior to the seizure studies, they were much easier to handle. So we determined, why don't we study the anti-anxiety effects of therapeutic ketosis? So we used a variety of things. The elevated plus maze, I feel, is the most reliable data. And that's like being on a catwalk. You can go out and be exposed to the environment, or you can run into the cave. So being on the open arm of elevated plus maze is sort of analogous to not having fear, having exploratory behavior. And we tested a variety of ketogenic agents, and the combination of ketone salt, sodium and potassium in equal concentrations, along with medium chain triglyceride, actually had the most remarkable effect. We thought it was going to be the ketone ester, but it was just simple medium chain triglyceride, caprylic triglyceride mixed with sodium, potassium, beta hydroxybutyrate, a racemic mixture, not the R enantiomer. This was a racemic compound. And we mixed that together, and it had remarkable effects, increased time in the open arm and decreased time in the closed arm, showing a reduced anti-anxiety effects. And in further studies, we looked at the adenosine A1 receptor, we looked at serotonergic, dopaminergic, and GABAergic. When we block the adenosine A1 receptor, it abolished that effect. So we think the adenosine A1 receptor is actually mediating some of these effects here, and maybe also the anti-seizure effects. So we're studying that now. So cancer is kind of a controversial topic. I know I'm running low on time, so I'll go fast with this. It's so much more, and you'll hear more about this tomorrow, I think, with Dr. Luke Cantley. It's so much more than just starving your cancer of sugar. And I get this all the time, that the ketogenic diet starves your cancer of sugar. It's so much more complicated than that, and so much more in depth. So that does happen. You decrease glucose availability to the tumor with a well-formulated ketogenic diet. You also suppress the hormone insulin, which decreases glycolysis and also IGF-1 signaling. But there's also a profound reduction in proliferation, in inflammation, in angiogenesis, invasiveness, and metastasis. We've published several papers showing reduced metastasis when in a state of nutritional ketosis. And that could be the ketogenic diet or ketone supplementation given with a standard diet in our animal model. And now we're working with different animal models on this, also a breast cancer model. So alter gene expression, enhance anti-immune activity. So an activation of the immune system may help your body identify and sort of neutralize cancer. So my colleague Adrienne Check at the Barrow Neurological Institute, she's at Arizona State Institute now, studies that. One of my students studies the anti-catabolic or anti-cachexic protein sparing effects of ketones. And I always thought from an evolutionary perspective, ketones, it makes sense that if we didn't make ketones, we would catabolize all our gluconeogenic amino acids, and we would quickly waste away. Our skeletal muscle would waste away. So ketones, we feel, we've demonstrated that they have pretty remarkable anti-catabolic effects. There's some evidence that acetoacetate can have an anabolic effect. So that's a whole project. That's my student's PhD dissertation he's working on now, and some interesting data coming out of that. And it seems to be, cancer cachexia seems to be driven by inflammation. So there's various cytokines that drive the muscle wasting, and we're trying to discern whether it's the ketones reducing the inflammation or the ketones having direct anti-catabolic effects. Also, an increase in the sensitivity of standard care is a big focus of what we're doing. Remember, using ketones as an adjuvant. And this is a paper here in Seminars in Cancer Biology that we co-authored with our colleagues, neuro-oncologists at Moffitt Cancer Center. And it really talks about targeting how the nutritional ketosis targets the Warburg effect. And you have a seminal paper is the hallmarks of cancer, and nutritional ketosis really targets, by changing your metabolic physiology, you are essentially targeting all the hallmarks of cancer. And that's really what the focus of that review article, that's our last paper we published. So this is more of a theoretical example here of what we're doing now. And I know there's actually patients and doctors implementing this right now, and kind of very severe patients that have really advanced cancer. But we're just doing the preclinical animal model work in different animal models, and we call it the press pulse therapy, where the idea here is to induce therapeutic ketosis to get a glucose ketone index between 1 and 2. That would be lowering your glucose to 3 and elevating your ketones to 3. So that would be a glucose ketone index of 1, right? So that has remarkable anti-seizure effects. I'm interested in that for brain cancer because brain cancer patients have seizures, and that was my original interest in using it for cancer, just for the anti-seizure effects. But when these patients have seizures, they're put on high doses of dexamethasone, and they're also put on anti-epileptic drugs. So I think we can eliminate that by just getting them into the therapeutic zone, and also using metformin and various exercises to get that glucose ketone index in the metabolic zone. And here's an example sort of what we're looking for, getting glucose down and ketones elevated at or above glucose levels. So when you're in that state, your body will be much more receptive to other modalities, including the standard of care, chemo and radiation. So the ketogenic diet, by inhibiting glycolysis, you're inhibiting the pentose phosphate pathway, which replenishes reduced glutathione. So you are crippling the cancer's endogenous antioxidant systems and making the cancer more vulnerable to other modalities, particularly modalities that kill the cancer cell through oxidative stress. So that's a big part of what we're looking at targeting. Hyperbaric oxygen therapy has 14 FDA-approved applications. Cancer is not one of them yet, but hyperbaric oxygen therapy is used for radiation necrosis. So patients who are getting hyperbaric oxygen therapy for radiation necrosis and following the ketogenic diet are getting some remarkable effects. And we have nice preclinical animal data. Intravenous vitamin C does not work by itself. It's promoted, but it does synergize with other therapies and also various metabolic drugs. So that includes lonidamine, a hexakinase inhibitor, hexakinase 2 inhibitor, 3-bromopyruvate, 2-deoxyglucose, metformin, dichloroacetate. So these are all drugs that we're studying by themselves, but also in combination with the other things that I'm talking about: ketogenic diet, ketone supplementation, hyperbaric oxygen therapy. So we're doing a lot of different things, and we're doing it in cell culture systems and also different animal model systems right now. So with just a short amount of time left, there are commercially available tools right now for assessing nutritional ketosis. I personally like the urine ketone strips, the Siemens Multi-Stick SG. So not only will it tell you your levels of acetoacetate, but it also measures 9 other parameters and monitors your kidney function. And also if you have like a urinary tract infection. So it's cheap, it's about 25 cents per strip. So it's giving you your ketone levels, right? But also telling you other information, and it's very cheap. So are you not in ketosis or are you in ketosis? So a lot of people kind of criticize the urine ketone strips. We use them for our research. The Clinitech status system is a device. If you stick the strip in there, it'll give you a digital readout of it. Breath acetone meters, I think, have a lot of potential. They're volatile organic compounds. We're interested in isoprostanes for oxygen toxicity, but I think there's a lot of potential in breath acetone. You will get a false positive with alcohol. Alcohol will give you a false positive, I found. Things like acetic acid, 2-carbon fatty acid, that's in vinegar. If you have a lot of vinegar, you'll be positive on these things. So that you do get false positives, but you need to sort of assess it, yourself. Chewing gum may give you a false positive too. Breath acetone correlates with seizure control. And in kids that do not want their fingers pricked, I think it's a very valuable tool. The blood ketone meter, we use the Keto Mojo. I think it's an awesome device. We also use Abbott Precision Extra. There are devices in the future that will monitor ketones, glucose, lactate continuously, like a Dexcom meter, like a Dexcom does for glucose. And I think really that's the future. So we're working with different people on that. So this is a summary of all the different projects we're doing. I think longevity is sort of something that we're interested in. We're keeping mice under different dietary protocols and looking at spontaneous tumor development. If we determine at day 200 that 30% of our animal model eating a standard diet has tumors, whereas the ketogenic diet, only 15% has tumors, that's a significant finding. And nobody's doing this kind of research. So we really think it's important to do longevity and prevention research. And we have a lot of projects with NASA on NASA Extreme Environment Mission Operations. And this is our lab, and there's a lot more people that we could add to that. We have a lot of undergrad students and pre-medical students as part of the lab, so we need to update that picture. And these are some of the resources. I compile all my resources on ketonutrition.org, and there's a number of publications here related to the ketone supplements I talked about.